Today we are going to know about Wnt beta-catenin signaling pathway. In this pathway, they determine the cell fate, determination, embryonic development, and cancer progression. This pathway has two states, the on and off, that is the active and inactive state. The Wnt off, the inactive state where no gene transcriptions occur. It consists of a receptor. It is known as the Frizzled receptor. Or we can say it as LRP5 or 6. It is inactive because of no Wnt ligand is present. Then beta-catenin is constantly degraded to prevent gene transcription. Then destruction complex, these are the names of the destruction complex: Axin 1 APC, GSK3 beta, CK1 alpha, phosphorylates beta-catenin. Then phosphopyrolated beta-catenin is recognized by the proteasome, leading to ubiquitinine mediated degradation. Without beta-catenin, TCF, LEF transcription factor remain inactive, leading to no gene transcription in the nucleus. Then Wnt On, that is the active state where gene transcription occur. Wnt's ligand bind to the Frizzled and LRP5 and 6 receptor, activating the pathway. Here the DVL protein is activated, inhibiting the destruction complex Axin, APC, GSK3 beta and CK1 alpha. Beta-catenin is stabilized and accumulates in the cytoplasm, and beta-catenin translocates into the nucleus and binds to the TCF and LEF transcription factor. This activates the gene transcription, leading to the cellular response such as the proliferation, differentiation and survival. So it is important in cell growth and development. It regulates the embryogenesis and organ formation. In stem cell maintenance also it is important to control the self renewal and differentiation. In uncontrollable cell growth, that is the cancer implications, in that also beta-catenin activations lead to diseases like colorectal cancer and hepatocellular carcinoma. This pathway is highly regulated, and its dysregulations can result in tumorigenesis. So we will see in detail this inactive and active states. As we all know the basic thing about this Wnt. First, the destruction complex formations takes place. Beta-catenin is bound by a multiple protein degrading complex containing Axin, that is a scaffold protein. Then APC. The full form of this APC is adenomysis polypsis coil and tumor suppressor. The glycogen synthesis kinase 3 beta, that is GSK beta. A serine and thionine kinase. Casein kinase 1 alpha, CSK1 alpha, another kinase involved in primary beta-catenin for degradation. Beta-catenin phosphorylation and ubiquitination takes place. CK1 alpha phosphorylates beta-catenin at serine 45, priming it for further modifications. GSK3 beta sequentially phosphorylates beta-catenin at serine 33, serine 37 and threonine 41, marking it for the ubiquitinine mediated proteasomal degradation. And E3 ubiquitinine ligase beta TRCP, that is the beta transducin repeat containing protein, recognize the phosphorylated beta-catenin and facilitate its degradation by the proteasomes. Then the TCS and LEF transcription factor remain inactive. In absence of this beta-catenin, TCF or the T cell factor, that is the TCF, and the lymphoid enhancer binding factor LEF, this both transcription factor remain bounded to the co-repressor, example HDACs and Grouch, leading to the transcriptional repression of Wnt targeted gene. The biological consequences of this is no activation of Wnt targeted genes takes place, suppressions of the cell proliferation and differentiation in the tissue where Wnt signaling is no required. It maintains the homeostasis in epithelial tissue and stem cell niche. In active state of the Wnt, that is the on state of beta-catenin stabilization and gene activation. Upon Wnt ligand binding, the destruction complex is inhibited, leading to the beta-catenin accumulation and nuclear translocation, activating the Wnt targeted gene. These Wnt ligand bind and receptor activations takes place. Wnt ligand secreted glycoprotein bind to the Frizzled receptor and low density lipoprotein receptor. It is known as the LRP 5 or 6 receptor. This low density lipoprotein receptor related proteins and co-receptor. These induce the receptor clustering and phosphorylations of the LRP5 and 6 by GSK3 beta and GSK1 alpha. Then the activated Frizzled receptor recruits Dysregulation DVL, a cytoplasmic signaling adapter protein. Then DVL inhibits the destruction complex by recruiting Axin to the membrane, disrupting the beta-catenin degradation. With the destruction complex inactivated, beta-catenin is no longer phosphorylate and escape degradation. Beta-catenin accumulate in the cytoplasm and translocate into the nucleus. In the nucleus, beta-catenin displaces the co-receptor from TCF and LEF transcription factor. Beta-catenin recruits co-activator such as CB5, CBP, CRB binding protein and P300 enabling transcription. This result in the expression of Wnt targeted gene involved in the cell proliferation, that is the cyclin D1 c-Myc differentiation and survival. The biological consequences of this is activation of cell cycle progression, that is cyclin D1 c-Myc, and stem cell self-renewal and differentiation, that is LGR5 and SOX9. The last is the regulation and termination of Wnt signaling. Since persistent Wnt activation can lead to cancer and several mechanisms regulating this pathway, negative regulation, for example DKK1, that is a DIC-COP1 and SFRPs secreted Frizzled related protein inhibiting the Wnt ligand binding to the receptor, and APC, that is the adenomatous polypsis coiling. This coil it is essential for beta-catenin degradation, that is mutated in colorectal cancers. Then Notum Wnt antagonistic, that deacetylates the Wnt ligand, reducing its activity. Then post translation modification, that is the E3 ubiquitin ligase, it promotes the beta-catenin degradation, and CK1 modulates the beta-catenin phosphorylation. Wnt signaling in diseases and cancer. In colorectal cancer, APC mutations prevent beta-catenin degradation, leading to consecutive Wnt signaling and uncontrolled cell proliferation. Then HCC, aberrant beta-catenin accumulate drive tumorigenesis. In melanoma, overexpression of Wnt ligand promotes invasion and metastasis. And in disorder and stem cell functioning also, it has an important role. Wnt signaling is essential for neuronal patterns and synapses formation. Mutation in LRP5 affect the bone density, that is the high bone mass disorder. The function of Wnt ligand is to secrete glycoprotein in which activates the pathway. Then Frizzled receptor, it the membrane receptor that initiate the Wnt signaling. Then DVL, it inhibit the beta-catenin destruction complex, and destruction complex namely AX1, Axin, APC, GSK3 beta, CK1 alpha degrades beta-catenin in absence of Wnt. Then beta-catenin transcriptional co-activator that regulates the gene expression. Then TCF or LEF, it consists of the DNA binding transcription factor, activates by the beta-catenin. Then CBP or P300 co-activators that affect the transcription, that facilitate the transcription. Then DKK1 and FRPs, Notum Wnt inhibitors that regulate the pathway activation. So Wnt inhibitor are being explored for cancer therapy, stem cell therapy, and used in stem cell therapy also it is used to control the Wnt signal for regenerative medicines. In Wnt signal role in immunology and Alzheimer and aging is an active field of study. Wnt beta-catenin pathway is fundamental in development, stem cell regulation, and cancer progression. Its precise regulations in the is for maintaining the tissue homeostasis, and its dysregulation in hallmark for various diseases. This was all about the Wnt signaling.